I'm Kathy Sakamoto, and I'm a pediatric hematologist oncologist. I treat children with blood diseases and cancer. This study focuses on a repurposed drug known as niclosamide to treat pediatric acute myeloid leukemia. Pediatric AML is a form of acute leukemia, which is the most common type of childhood cancer. It represents about 25% of acute leukemia patients in children, and less than 60% of children with AML survive. There are very few treatment options for children who relapse, and less than 20% of them survive. The treatment for pediatric AML causes hair loss, weakened immune system, and many months in the hospital. Oftentimes, children cannot go to school, play with their friends, or do their normal activities because of their treatment or complications from their treatment. AML represents 700 cases or 8 million children with AML diagnosed per year. It is a rare orphan disease, and drug companies are not interested in developing new drugs for pediatric AML. If our approach is successful, we can use this globally for over 29,000 adult patients per year. The current treatment for pediatric AML is intensive chemotherapy, which are literally poisons and have many side effects in growing children and also late complications in survivors, which impacts their quality of life. The treatment is expensive, with intensive chemotherapy or stem cell transplantation costing somewhere between $500,000 to $1 million. Current therapy for relapsed AML have limited to no efficacy. And new therapies, such as immunotherapy, will not be available for at least 2 to 3 years for pediatric patients. The proposed treatment involves targeting a protein known as CREB. Our lab found that CREB is overproduced in AML cells, and this is associated with a worse prognosis. Therefore, we developed a small molecule to target CREB and inhibits its function in collaboration with a colleague at Oregon Health Sciences University. Unfortunately, this molecule is not ready for human use. So in collaboration with a medicinal chemist at Stanford, we identified an FDA-approved drug known as niclosamide, which, as you can see, has very similar structure to the small molecule CREB inhibitor. Niclosamide is an anti-tapeworm drug that's FDA approved and remains on the WHO's list of essential medications. It was withdrawn from the US market in 1996 due to low usage from better drugs for parasites. Interestingly, niclosamide has also been recently reported to potentially inhibit SARS-CoV-2 or coronavirus. Niclosamide is well tolerated in children, and it's not toxic, with the only reported toxicity being GI. Now, niclosamide in preclinical studies has been shown to inhibit cell growth in culture of AML cells. It decreases CREB activity and doesn't affect normal blood cell production. It importantly suppresses AML progression and increases survival of mice that take niclosamide every day in my mouth. Niclosamide also works together well with chemotherapy drugs to enhance killing by chemotherapy drugs such as cytarabine. Our study is a phase 1 clinical trial for relapsed refractory pediatric AML. The primary objective is to study safety in a recommended phase 2 dose of niclosamide in combination with the chemotherapy drug cytarabine in patients with advanced myeloid leukemia. The secondary objective is to evaluate the pharmacodynamic effect of niclosamide in myeloid leukemia cells with or without cytarabine. In other words, is the drug hitting our target CREB? And finally, the exploratory objective is to evaluate the pharmacokinetics of niclosamide in children with relapsed refractory AML. This is a clinical trial that involves dose escalation of oral niclosamide once or twice a day for 14 days, followed by 5 days of cytarabine. Patients who are over the age of weight of 34 kilos and age of less than 25 years are eligible. The sample size is approximately 16 patients to evaluate 3 patients per dose level during a 2-year period. The budget for the entire clinical trial will be approximately $1 million. However, we have additional funding from the Leukemia and Lymphoma Society and Cure Childhood Cancer to support this clinical trial. However, we do need additional funding for pharmacokinetic and pharmacodynamic studies, data analysis, and salary for a biostatistician. The team at Stanford includes a clinical team, including myself, Dr. Norman Lacayo, who is Section Chief of Oncology and Director of the Poetic Consortium, Eileen Monge, who is our pediatric research nurse, and our research team includes Kara Davis, who's Assistant Professor of Pediatrics, who will help us with the pharmacodynamic studies, Adam Fry-Moore, who is our clinical pharmacologist, and Haley Hedlund, who is our bioinformatician, who will perform the data analysis. The impact of our study is high because our clinical trial will potentially provide a non-toxic, effective treatment option for children with relapsed refractory AML and allow us to go into phase 2 trials. It could potentially improve the survival and quality of life in our patients since the drug can be taken at home. The treatment is inexpensive, $9 per pack, which is 4 tablets, compared to $500,000 or $1 million for intensive chemotherapy or transplantation. And if niclosamide proves to be well-tolerated and effective, this would globally impact approximately 6,000 to 7,000 relapsed AML patients per year. So our clinical trial has the potential to change the lives of many children and adults with AML. Thank you for your attention, and I'm happy to answer questions.